Well, I'd like to thank Joshua Weinstein and Eric Hollander for inviting me to speak with you today. And I want to congratulate them on the great work that I Care for Autism has been doing. It's really remarkable. So today I'd like to cover some information about an important syndrome. We think it's the leading cause of inherited developmental disability, intellectual deficiency. And it has a lot to teach us about autism, we think. So I'd like to cover some of the features of the syndrome and how it relates to autism and what we can learn from it. The condition affects males primarily because it's X-linked. It's on the gene mutation is on the X chromosome. So since males have one X chromosome and females have two, it affects males more than females. And it runs in families. And as I said, we think it's the most common inherited cause of intellectual disability, and it's due to the silencing of a gene, the Fragile X FMR1 or Fragile X mental retardation gene 1 on the X chromosome. And this gene makes a protein, and the protein binds to RNAs, to messenger RNAs of other genes. So it modifies or modulates the expression of about 5% of expressed brain proteins. And it is a family disease, so it runs in families in a similar way to hemophilia in the British royal family. It's X-linked. But it's kind of semi-dominant. You can carry it be a carrier in the male and not express it, but the gene undergoes an expansion with generations. And I'll explain a little bit more about that. So historically, it's a relatively recent syndrome to have been identified. It was only really characterized recently. The first family was published, we think, back in 1943. It was a large British family and was known as the Martin-Bell syndrome. In 1969, Lubb saw a family with a Fragile X chromosome and wrote it up as a paper, but it was kind of forgotten about, and I'll show you a picture of that. But in 1977, Grant Sutherland in Australia was studying marker chromosomes, fragile chromosomes, and he discovered that when he moved from one lab to another, he couldn't replicate it. And that led to the finding that you had to have low folic acid in the culture to bring out the Fragile X chromosome. Then Gillian Turner looked at a series of male brothers and found that close to half of them had this fragile marker site. And those are pictured here. And this was the family that Herb Lubb saw, a pair of brothers, and shown when they're young and as they got to be older. And they came from a pedigree where there were 4 or 5 male relatives, and they were all related through their mothers. So this was the first chromosomal finding that related to this marker. And the name comes from the tip of the X chromosome. When the gene mutation is present and the cells are grown in low folic acid medium, we see a stretching or a breaking near the end of the X chromosome, and hence the name Fragile X. So the first 2 brother pairs that I saw are shown here, and they have some of the typical features that we've come to associate with Fragile X. They have somewhat large ears or long ears, and they have kind of a long, narrow face, particularly this brother on the right, a little bit of a dysmorphic appearance. So this long face tends to get more prominent with age, and they tend to have a narrow palate. This boy particularly had a very high arched palate. And here was another family I saw early on. There were 3 brothers that were all Fragile X positive, and they were all fairly severely affected. This boy was 10 years old, 11 years old, and he had both a severe form of autism and very low functioning. So he couldn't be coped with at home, and his mother had to have him placed. And here's another man that I'd met early on. Well, I met him when he was 65, but shows pictures of him developing. And as a baby, he didn't look particularly different. But as he got older, he had a longer, narrower face and big ears. He passed away at the age of 85 a few years ago and had a really lovely personality. And we found the Fragile X chromosome mutation to be present in most ethnic populations and probably relatively similar frequency. About 1 in 4,000 boys are born with the syndrome, and about 1 in 200 women are carriers. And here was a boy from Beijing, China that I got to meet, and it had large ears. So the syndrome is characterized by this moderate to severe degree of retardation with an IQ in the range of 20 to 60 being typical, with a long face, high prominent forehead, and prominent jaw. They also have very prominent ears and loose jointedness and typically flat feet. And often they can touch their thumb to their arm, showing the degree of flexibility. And they don't seem to have any degree of Alzheimer's as they age. They may get a little bit more aggressive at times, but we haven't noted any particular life-shortening effects of having the mutation. But not everyone with a long, narrow face and big ears has Fragile X. Here's another boy showing it as he gets older. And having big ears is typical of some dwarfs. Dopey, I think his name was. One of the unique features or unusual features is to have a large testicle volume. And here was a male with a macroorchidism. We plotted out the testicle volumes that we measured on the first 50 or so families, males we saw. And you can see that the typical size is in the range of around 50 mLs, and the normal average adult male is about 17 mLs with a range up to about 25. So this is a prominent early sign that develops during adolescence. So just to kind of summarize the phenotype, long ears, long face, large ears, an average IQ of about 45 with a range up to 80, speech delay, deviant language, hyperexcitability, and behavior problems. And as I'll get into, there's a strong association with autism. The IQ distribution early on, this was from a paper from Grant Sutherland where they looked at institutionalized males that had been identified, and they found that most had an IQ in the range of 20 to 50, but that doesn't include children. Looking at children, we've found that going from age 5 to about 12, there's a decline of IQ from a normal range of 80 down to about 50. And this is plotted out here. So 45 to 50 is the adult normal IQ that is measured. And in addition to the intellectual disability, the language abnormalities with a lot of perseveration and tangential speech, a lot of anxiety, attention deficits, sensory integration problems, auditory processing problems, and hypotonia are noted. One of the early behavioral features that was commented on is this lack of eye contact with, and here was a picture showing the typical response of a male to a visiting clinician. And even brother pairs have this unique greeting ceremony where they don't look each other in the eye. Now, it does also affect girls. If one of the two X chromosomes has a mutation, it can be expressed in the girls as well, but it's usually milder. Among affected females, about 50% are intellectually deficient with an IQ below 70, about 25% in the range of 70 to 80, and 25% are normal, but often they also have some behavioral problems such as shyness, anxiety, and low self-esteem. I'll move on now to the brain. The brain in Fragile X grossly looks quite normal. There have been some subtle features described. Alan Reese described that the vermis of the cerebellum was a bit hypoplastic, but it's not a very characteristic finding. But on neuropathological analysis, we were able to look at 3 males early on and pointed out that the dendrites looked like the spines were abnormally immature. They were long, spindly, rather than short, stubby, as is typical for more mature spines. And this kind of highlights that difference. The normal spines become short and stubby, whereas Fragile X seem to be immature, long, and spindly. And this has been replicated a number of times. Now let's look at the molecular genetics and molecular mutation. So the mutation affects the Fragile X gene. There's a repeated sequence of CGGs. The normal number is close to 30, but ranges from 5 to 55. Once it is inherited in a range that expands, it tends to grow from generation to generation. And this leads to the gene getting turned off, and it's the lack of expression of the gene that causes the syndrome. Rare patients have been seen, about 1% or less, who have deletions or point mutations within the gene. And this is a picture of the gene where the repeat structure is shown here upstream or in the promoter region of the protein coding part of the gene. And the normal range up to about 55 repeats. Premutation is the carrier state, where between 55 and 200 repeats, the carriers of these premutations, mothers or fathers even, are basically normal, though there are some late onset effects, and we'll describe a few of those in a minute. The full mutation occurs when this is inherited and then it expands to greater than 200 repeats. It undergoes a methylation process, and that turns the gene off. And that's the underlying cause of the gene defect. So the repeat region can be analyzed by molecular means using an enzyme that cuts around the region and having a probe, and then using a second enzyme that's methylation sensitive. And when that is used, we get a pattern of bands that's different than if the region can't be cut, so we get a long region. And this is shown on a figure here, a typical southern blot, where a typical male has only one band that is cut by both enzymes at 2.8 kilobases. A normal female has a normal active X and a normal inactive X that gets cut. And then carriers have two bands of different sizes, and the mother here is, the daughter has a large band, and then the affected children have large expansions in the range of greater than 200 repeats. We also see some individuals that have a mosaic pattern, that have both a full mutation and some degree of premutation. And we call this size mosaicism. And we've looked at it carefully and find that 20 to 40% of males have this mosaic pattern, whereas there's also some males that have unmethylated, partially full mutation. And these are shown here on some figures of southern blots of mosaic patterns. Now, mosaicism, we found, predicts somewhat degree of functional outcome. These were Vineland scores, and these individuals were Fragile X with a mosaic pattern, and they generally did higher and were more functional than those that had only the full mutation. We divided a group that we'd found that had autism as well, and that didn't predict very much about the increase, though there was a slight increase. Now, the repeat is interrupted by an AGG, and that tends to stabilize the inheritance of this region of the repeat. Whereas once it starts to expand, it loses this AGG, and that makes it more unstable in inheritance pattern. We developed, once the gene was cloned, we developed a PCR method to allow rapid amplification and rapid detection of the mutation. And that allowed us to, using special techniques, see full mutations by PCR as well as the normal size range. And it allowed us to document the range of normals as well as the larger size premutations. Looking at, we looked at 2,500 New York controls, seeing that the most common repeat number was 30, followed by 29, and then 31, and 23, and 20. And about 0.5% had a repeat that was greater than 50. So we have come now to recognize that the normal range is up to about 55. We call this this intermediate range 45 to 55 repeats. And carriers are in the range of 56 to 200, and the full mutation is when it expands to greater than 200. Now looking at this, we can predict how common are females as carriers. And this has been borne out by studies of populations that about 1 in 150 women have a repeat that's greater than 55, and 1 in 300 greater than 60. And and that's something in the order of 1 in 200 to 1 in 275 are the current estimates of the carrier frequency in the U.S. population. The gene has this triplet repeat region near the promoter region, within the promoter region, but it's transcribed, but it fails to get translated. There are also some isoforms at alternate splicing, but we're not yet sure that they're of any great significance. The gene encodes a protein. The protein has RNA binding sites, 2 KH domains, and an RGG box. These attach to messenger RNAs. And particularly about 5% of brain messenger RNAs are bound by this protein. And we think the main function is to modulate RNA translation. Now, what are the genes that it binds to? Well, many of the genes that have been, the proteins that have been found to bind to it are genes that are active within the synapse, or synapses. And it primarily downregulates their expression until the right signal comes along. Now, recently, Jennifer Darnell, who's at Rockefeller, did a very sensitive test to find out what the proteins were using a CLIP assay, and found that she identified 842 target messenger RNAs that the FMRP would be attached to. And then she analyzed the Simon's Safari database of candidate autism genes, which is something over 100 genes that have been annotated to be probably associated with autism, and found that the overlap was about 1/3. And many of these are involved with synaptic activity, and many are being studied individually for their relationship to autism. So we can see that there's an overlapping target of the Fragile X protein and these various genes that have been identified as having contributed to autism phenotypes within some families. And this figure from one of their publications shows in blue the various targets that the Fragile X protein attaches to and modulates. And when it's missing, then these are not properly regulated, and it leads to synaptic dysfunction and synaptic immaturity and altered functioning. It's been studied by Carolyn Smith and her colleagues at NIH that when you're in the mouse model of Fragile X, when they're missing the protein, they overexpress protein, these general proteins. So here's the normal control or wild-type mouse showing a cross-section in the hippocampus region. And here's the knockout mouse showing that the level of protein is higher. It's hard, it looks more red in both the hippocampus and in the core of the brain. And again, here's the control and the knockout showing overexpression of about 20% more protein. So there's a dysregulation of protein synthesis. Donato Loesch from Australia, along with Randy Hargeman, did a study of quantitating protein by an assay they had developed to look at the range of amount of FMRP and IQ on this chart here. And they came to the conclusion the more Fragile X protein you have, the smarter you are. These were looking at both full mutation males and mosaic and pre-mutation carriers, as well as controls. So lacking FMRP is not healthy. Now turning to autism. We reported back in 1982 that it was associated. We found 5 of the first 22 males with Fragile X that we identified had been previously diagnosed with autism, or 23%. And that led to various people looking at their population of Fragile X as they became more and more identified, and the overall mean was about 25% to have autism, but the range was some 7 to 60% in various studies. Looking at autistic males for how common is Fragile X, we undertook a study, a multi-site study, and accumulated 183 patients with autism that we screened for Fragile X, and we found 13 that were positive, or 13%. And then the various other studies that have been carried out showed an overall mean of about 7%. But these were done up to about 19 90 to 2000. We conclude that many children with Fragile X have autism, and I'll get to the data on that in a minute. And perhaps 1% to 6% now currently have Fragile X in various subjects and various studies. This was a chart that was published by Harris and Hagerman, where they did ADOSs on a series of 47 boys. And they found that with a cutoff range of 12, that close to half were either had autism or PDD and OS by the criteria at the time, and showing that there was a gradient effect. But that 50% figure has been replicated a number of times by other groups now. Now, the behavioral characteristics of Fragile X males in many ways are similar to autism. There's perseveration, there's stereotypic behavior, problems regulating affect, attention problems, and hyperactivity. We think in general anxiety is probably more common in Fragile X, such as the gaze aversion, whereas their social interests and perception of others and attachment to parents are higher in Fragile X. The Cure Autism Now was a foundation that was set up by two Hollywood producers, and they had a son with autism, and they founded an organization that was involved with funding grants for autism research. And I joined the group as one of their scientific directors, and their goal was to obtain families where there were 2 or more children in the family that had autism in order to highlight and help identify genetic factors that might be involved. And we undertook to screen the probands in each of the families. And in the first 480, we found that there were 6 that had Fragile X, but we found that there probably were about a number of the families that had had prior testing. So we calculated that about 2% of this population early on was positive for Fragile X, and indicating a significant association. But it was lower than the 7% or so, 4 to 7% figure that we had expected. And what was the reason? Well, the IQ of the autistic agree subjects turned out to be about 80 on average, with a range of 34 to 144, whereas the IQ of the typical Fragile X was on the range of 45. So that can be explained by the changing diagnostic and the increasing frequency with which we see individuals on the spectrum that are not affected or have an IQ above 70. And in fact, that's consistent with California data, as well as the large CDC study, where around 40% have an IQ below 70, so 60% are above. And that's also consistent with the CIMAX collection that includes the average mean IQ is about 90. So I'm going to skip on a bit here to kind of conclude. We've found that in the mouse model, it was found that the glutamate pathway was overexpressed, and that led to the development of some treatments by Roche and Novartis and Seaside Therapeutics. These were mGlu-R5 antagonists, and these were carried out in fairly large-scale trials. Now, unfortunately, they didn't do better than the placebo, so they were stopped. But there have been ongoing studies of other drugs, medoxidine and an IGF-1 drug is in trials currently. There are others that are being proposed, and I just heard that the NIH is funding a childhood study of the Novartis drug that should get underway in the next year. We know that a number of genetic syndromes have an association with autism, so by studying specific genes, and we might learn a lot more about underlying factors related to autism. And in fact, I'm going to be attending in a couple weeks a Gordon conference that's focused on Fragile X and autism-related disorders, hoping to come up with new insights into disease mechanisms that could lead to new therapeutics for neurodevelopmental disorders, for Fragile X, as well as for some individuals with autism. And so I hope we're moving in the right direction, and I thank you for your attention and for my collaborators. Thank you.